Started this morning.
Yeah.
So for people that don't know me, my name is Peter Westley. For the last few years, I've had the total pleasure of working with Maddy. And as an advisor, defense days they're really full of emotion. So it's like bittersweet, it's exciting. It's the start of something new for Maddy. It's the end of something for me in the lab. But the best part is I get to give an intro and show pictures and do a little bit of a journey. So I looked back last night, June 4th, 2020, right when pandemic is rolling. I get this email from this student, laid out, very elegant, I want to do this project. Here are all the partners I have lined up. This is what I've done. I want to make it happen. Are you interested, essentially? And I wrote that back, yes, I'm interested. And away we went. So that was June 4th, 2020, and here we are. So it's been really fun to watch Maddy grow as a scientist, grow as a person. So they students that kind of go through this journey where they start, let's see if I can awkwardly advance this. There we go. So they start a little bit confused. Things are all new and shiny, haven't been used much. They're not really even sure how things work. You need water, but you watch them really mature they start simply, so it's still pretty shiny, but there's not much in the boat, but you're on the water paddling to the point where you start getting loaded down with things, looking happy. And I think there might even be some dead animals in this boat. So you kind of see this progression of learning that occurs. And that's been really, really fun to watch. They also learn that you don't have to cheat by holding out the fish to make them look big. I mean, you catch big fish. So if you catch a big fish, you don't have to hold it out that far. We've had that conversation, and they learn. And then you can see the smile is sort of commensurate with the fish there. You're missing some of that with her behind those things here. There she is. Okay. So what else? But she has also become part of this really amazing group of what I would refer to as, wait for it, badass Alaska women. So she has learned the joy of walking in wild places like the tundra in the fall, not just for a walk, but walks are better when you have a rifle on your back looking to harvest something. So she's discovered the joy of that. Catching fish with friends, eating snacks above tiny cheese above wild places, and ridding the world of these evil grazers that are up there, thinking about the moss and the grass that are up there, saving those environments from these things with friends. So that's been pretty amazing to watch. But she's also grown a ton as a scientist. So Maddy learning that tags don't go in your mouth, they actually go on the fish. So she's figured that out. And putting together this research project from scratch, driven by her ideas, melding it, sort of growing a kernel of an idea into something that was really special. That's been really fun to watch, Maddy. And I don't know what's coming next. I won't ask, what are you doing next? I know that's awkward, but I do know that whatever you do, that you will show up fierce, impeccably dressed, and ready to build fires and bring the heat, whatever you do. Okay, so anyway, so the stage is all yours, Maddy. No, I'm not going to give away my sources. We never tell. They may or may not be in this room. Have fun. Okay.
All right.
Yeah.
The smize is real.
Wait for it.
Hopefully everyone on Zoom can hear us still and
You're probably
are
reminding
with
me
us.
all the time.
How many people are on Zoom? 47? Dang.
The world wants to know. Okay, stage is all yours, Matthew.
All right. Well, thank you everyone for being here. This is really a special moment for me, and ultimately this is like the culmination of 3 years of work, and so I'm I'm just really grateful to be sharing this with you all. Peter's been an incredible advisor, and my steering committee has been integral to the process. Vanessa Von Vila, Steve McCormick, and Andy Seitz. They couldn't be here in the room with us, but they're on Zoom cheering us on. Welcome. And so this is my master's in fisheries thesis defense. My research topic is looking at the patterns and variation of heat stress expression and spawning Chinook salmon, and linking that to their reproductive success and potential consequences associated with warming temperatures. All right. So I just want to take a moment to acknowledge that I did do my research on the Ninilchik River, which is the traditional and current land of the Ninilchik village tribe. And I had the pleasure of working with Greg a little bit and learning about the history of the river and just how much the fishery has changed over the years. And it's just been an honor to work with the Chinook salmon in the Ninilchik River. They're just really majestic creatures, and that wouldn't have been possible without the land stewardship of the indigenous people in this region. So I just want to give a broader overview of my thesis before we dive in here. So for my chapter 1, I'm looking at variation in heat stress expression in migrating Chinook salmon, comparing between 2 different rivers here in Southcentral Alaska. And then for my second chapter, I'm really trying to look at the reproductive consequences of having heat stress in these salmon. So just taking a step back, why am I doing this research? Pacific salmon are incredibly important for the state of Alaska, but ultimately for the entire world. We really rely on these, we really rely on these Pacific salmon, and it's just really critical that we understand how warming temperatures are going to impact them moving forward. And as we've seen over this time series on the x-axis, 1974 to 2018, and on the y-axis, the deviation from the average. This is taken from Alaska Department of Fish and Game. And we can see that there's been below average returns of Chinook salmon since about 2007. And so there is major concern as to why these Chinook salmon are declining in Alaska. So USGS has done research on the Yukon River looking at heat stress. And this is Vanessa Von Vila's work in 2020. And they did find a prevalence of over 50% of the fish sampled had heat stress. And this was looking at a known biomarker, HSP70, which is heat shock protein 70. It's a protein that's released in all living organisms, and it actually protects our cells from damage when we experience our upper thermal limits. But too much of this protein can also be really energy intensive, and concerns of Chinook salmon expressing too much HSP70 will be explored in this paper. Again, Von Vila's work and Donnelly et al. 2020, they found that there were these thermal thresholds that Chinook salmon on the Yukon River were experiencing. So at 18 degrees Celsius, we're starting to see the first level of thermal stress. And then at 21 degrees Celsius, we're starting to see the upper limits of stress where fish were actually dying in these tanks. So then looking at all these different factors together, we can start linking that to the potential reproductive success of migrating Chinook salmon on the Yukon River, specifically as like our case study to then expand out to the rest of Alaska. So certain climate drivers are longer migrations that are increasing their need for energy to make that route all the way into Canada. We also have poorer marine foods in the oceans now, so less metabolic stores for when they make that long migration. And then the concern that we're focusing on in our research is higher water temperatures, potential for heat stress, which has been then linked to reduced reproductive success, so premature mortality. And so that looks like when you have pre-spawn mortality, the eggs are still retained in the salmon. You can see that on the picture on the left where, there's the fish has died without spawning fully. So this was a major concern in 2019 when we had this major heat event. And there is documented pre-spawn mortality across the state. Peter Westley and Von Biela has published papers showing these pre-spawn mortality events, such as in Prince William Sound on the right. You can see this, these fish didn't have a chance to spawn and the rivers that they usually travel up were dried up. So this is really concerning for salmon because they're semilparous, and so they only have one shot to spawn fully. And when they die before doing so, it's kind of like all their eggs are in one basket and they lost that chance. So it's devastating. So South Central Alaska, where this study is located, is a glimpse of how fish are going to respond to these pressures that are happening on the Yukon as well as other parts of the state of Alaska. So we're having increased anomaly temperatures that are above average. We're also having greater human activities. About 50% of the state's population lives in Anchorage, and a lot of them move down to or come down to the Kenai and the Ninilchik River to fish. And so there's a lot of sport fishing pressures on these systems, as well as just people coming down and developing along the river. So declining Chinook salmon runs has ultimately led to greater hatchery reliance. So looking at Sue Mager's paper in 2017, we can see on the x-axis the weekly max temperatures, and on the y-axis a range of rivers here in South Central Alaska. And we can see how climate change is currently and projected to increase the water temperatures of our local rivers. So the dashed line at 18 degrees Celsius is that initial thermal stress of Pacific salmon, and the 18 degrees Celsius or the 20 degrees Celsius is the upper thermal limit. And you can see that about 2/3 of the rivers located in South Central Alaska are either currently or projected to be much above that threshold for stress in salmon. So hatchery Chinook salmon are a major economic driver in this area. We have the fishing hole on the spit here that's stocked with hatchery Chinook. I participated in the fishery, and it's big reason why people come to Alaska. And it's an important fishery that we have. And so, but there are trade-offs with having hatchery Chinook salmon. There's concerns of reduced genetic diversity in the wild populations with increased hatchery strays. And so we need to start thinking about what's the implications of having so many hatchery fish that we rely on here. And so more specifically for this research, I'll be considering the juvenile rearing stage between hatchery versus wild fish. Hatchery fish are reared in a facility, and so they have different upbringing or temperatures that they're raised in or reared in, and so this could affect their thermal tolerances as they become adults, which we'll focus on in this study. So salmon in this changing world, they really have three options. They can move, they can go further north to cooler temperatures, they can adapt, and that's really what we're focusing on in this research, is understanding, the potential for thermal plasticity, as well as, the variation that's associated with heat stress expression and how that variation could be the beginnings of adaptation even in these regions. So how are they going to adapt? And then worst case is whether they would die from too much high temperatures in these systems. So to go through my first chapter, I broke it up into research questions, again, looking at the patterns and variation of heat stress. And For my first question, I'm looking at the difference between heat stress expression between two populations, Crooked Creek and Ninilchik River. Then next, I look at the rearing conditions and how that may differ their heat stress levels, so hatchery versus wild. Comparing body sizes. So I took measurements from the mid-eye to the tail fork of the salmon and looked at differences of heat stress expression. And then whether or not they were held or not held as part of the hatchery broodstock program, which is how we create more hatchery fish. So when I refer to broodstock, I just mean fish that are mature and ready to be artificially spawned or spawned by people to create more hatchery fish. So just to give some broad overview of my chapter 1 and with each of these questions, what led me to potential hypotheses of what I'm going to see in my results. We know that fish populations can adapt to their thermal environments and have site-specific thermal tolerances. And so Eliasson's paper in 2011 has found these population-specific thermal tolerances. And that's really due to, potentially acclimating to their environment. So Pacific salmon are getting used to these increasing temperatures. They could be so acclimated to that environment that they're actually having less sensitivity to these warmer temperatures. We also know that fish reared in the wild will have different reactions or may have different reactions to warming temperatures in their adult life stage than fish that are reared in the hatchery. And so hatchery fish are reared in a pretty stable temperature, usually a little warmer to accelerate their growth. And wild fish have much more variable temperatures, and this can create more more variation in their thermal experience as they are adults and are more accustomed to that thermal range as juveniles. So a study has come out in showing that there's actually greater HSPs in wild fish than hatchery fish. So we expect to see that as well on this system. Studies have found that body sizes of fish, well, there is potential that body size could influence heat stress levels, but the studies that we found were that there was a decrease in thermal sensitivity with smaller fish because of their metabolic needs are less, and so they may be less sensitive to those changing temperatures. So studies have found that holding and handling may not necessarily increase HSP 70, but it does increase cellular stress. And so looking at the broodstock holding facility at the Ninilchik River, it's a small raceway, there's lots of fish in there, so it can be a stressful process that fish undergo. And so although this isn't temperature related, we think that there is some other factor other than temperature that's impacting the HSP 70 levels of fish held as broodstock. And so that's ultimately some sort of additive effect here of just multiple stressors at play that could increase those levels. And I guess I skipped over that. For those who are still a little confused about what broodstock are, they're just mature fish that we hold and are artificially spawned for hatchery propagation. So this research was conducted on the Ninilchik River and Crooked Creek. They are two rivers that are about 40 kilometers apart, located in Southcentral Alaska. And Crooked Creek is actually a tributary of the Kasilof River, which is a glacial fed system. And so it has on average cooler temperatures than say the Ninilchik River, which is groundwater fed, more subjected to ambient air temperatures. And so both of these rivers have segregated hatchery programs, which means that they have wild and hatchery fish that are that return each year, but the hatchery fish are removed. So there's no spawning with wild fish on spawning grounds. It's a little hard to see, but we collected water temperature data, or Cook Inlet Keeper collected water temperature data, where my cursor is located here on the Ninilchik and then up here on Crooked Creek. So relatively close to our sampling sites. And what the water temperature data was showing us was what we suspected, that Crooked Creek, which is in blue, Ninilchik River in red. Looking at this plot, the x-axis is our seasonal temperature data for when Chinook salmon are entering these systems in late May. And on the y-axis is the daily max temperature in Celsius. So we also have that 18 degrees threshold for stress labeled. So you can see that Ninilchik River is reaching above that threshold for stress while Crooked Creek is staying below it. And that Ninilchik River is warmer than Crooked Creek. And then looking at 2021's data, we only collected samples on the Ninilchik River for that year because we were more focused on the hatchery program there, as well as because it had warmer temperatures. And so you can see that it reaches above that 18 threshold in 2021 as well. But on average, both of these years are relatively on the cooler end. We're not seeing prolonged levels weeks on end above 18 degrees Celsius. The methods that I used to collect heat stress quantifications was a muscle biopsy that was non-lethal, and it just took a little tissue sample that was sent to a lab on the East Coast with USGS. And we collected samples on both Crooked Creek and Ninilchik River. And yeah, it was a relatively easy way to quantify heat stress in spawning Chinook salmon. The methods that I used to try to understand my first and second objectives in my chapter 1 was first modeling an overall generalized additive model, which included a bunch of terms such as the temperature interacting with rearing. So rearing is whether or not it was a hatchery or wild. The temperature metric I used was past 5-day max temperature. I also included an overall river term, which is whether or not the fish was collected from Crooked Creek or Ninilchik River. And then again, another overall rearing term, so hatchery versus wild, as well as smooth size terms, which is the body size measured in millimeters, and then the day of sampling that the heat stress sample is collected to understand whether or not the level of maturation that these fish underwent was impacting its heat stress levels. So the response variable here is HSP70. And I used a stepwise regression approach, which means I found in my model the highest p-values and removed them until I found a reduced model that had significant p-values. And I also included smooth terms because it is a GAM model, so it needs to be more flexible to accommodate the heat stress data that has a threshold. So the best reduced model that I found was including temperature and rearing as an interaction term, river, so Crooked Creek versus Ninilchik River, and then rearing and overall hatchery or wild effect here. And so you can see that plotted on this figure. On the x-axis, we have past 5-day max temperature in degrees Celsius, and the y-axis, we have HSP70 concentration. And so we can see that Crooked Creek on the left in blue actually has higher HSP70 expression than Ninilchik River in red, which is a little surprising. But again, thinking about being a cold water adapted, and so maybe a little more sensitive to those warmer temperatures that are below like heat stress levels, which is above 18 degrees Celsius. As you can see, the Crooked Creek never actually even hit above 18 degrees Celsius in our study. And you can also see from this plot that wild fish, which is the solid line, had greater heat stress expression than hatchery fish in both rivers. You can also see that on the Ninilchik River, we have temperatures that surpass the 18 degrees Celsius threshold. And you see a trend of much higher heat stress levels. So you can only imagine what it would have looked like if we did have those prolonged levels of temperatures that were above 18 degrees Celsius. And then to look at the effects of broodstock handling on heat stress levels, I then modeled another GAM model. And then my terms were whether or not broodstock was held or not held for propagation. I then included it as an interaction term with temperature again, and then I included it as an overall effect, as well as a hatchery versus wild effect. So that's rearing. And then again, body size and day of sampling as terms to look at in the model. I used a stepwise regression approach, and the best reduced model that I found was that temperature and broodstock had an interaction and had an influence on heat stress expression. And so you can see this plotted here on, again, x-axis past 5-day max temperature, y-axis HSP 70 concentration. We're seeing that for the fish that were not used as broodstock, there's a pretty normal trend with temperature and a normal response as it interacts with temperature. But then for the fish that were not used as broodstock, there's this pretty significant uptick of heat stress levels that are at 16 degrees Celsius, which definitely signs that there was some other stressor involved that resulted in these higher heat stress levels prior to what we would expect thermal stress to occur. So just to recap my chapter 1, how does heat stress differ between two populations? We found greater heat stress in Crooked Creek, the cooler population, than Ninilchik River, the warmer one. We found a hatchery versus wild rearing effect with greater heat stress in wild fish than hatchery fish. We also found that body size and day of sampling had no effect on heat stress. And so this implies that for the maturation stage that we were thinking could be resulting in greater stress in fish that are held as broodstock, because usually they're further along in their maturation stage. Since it had no effect on heat stress levels, then we really think that there is potential that the broodstock holding is increasing the HSP 70, but it's not temperature related. So it's not increased water temperatures driving it. So now moving into my chapter 2, which is looking at the reproductive consequences of heat stress expression in spawning Chinook salmon. My first question is whether or not heat stress differs, or heat stress in spawning females affects their egg retention on the wild spawning grounds. And then whether or not egg survival to the eyed stage in the hatchery is affected by heat stress levels. So we know that the spawning adult and embryo life stages are the most vulnerable to these high temperatures. And so that's why we focused on spawning adult stage, as in looking at egg retention as the heat stress in the spawning adult stage and how that pertains to egg retention on the wild spawning grounds, and then egg survival to the eyed stage. So looking at the embryo life stage there. We know that there are trade-offs between expressing heat shock proteins and reserving that energy to spawn successfully. So HSP 70 is awesome to, protect when it's really hot. We know that it protects cells from damage, but too much of it could be taking energy away from these really important reproductive processes like maturation and gamete production, as well as just defending their nests and having the energy to spawn fully on spawning grounds. So we know that higher spawning temperatures can actually drive lower egg survival. Looking at heat stressed females prior to their spawning, they actually found that it can result in altered ovulation timing and even an inability to ovulate at all. And then in a worst case scenario, just increased developmental abnormalities of these eggs. So looking back to my chapter 2 and looking at the first question, whether or not heat stress impacts egg retention on the spawning grounds, I used esophageal radio tagging to be able to identify the spawning grounds on the Ninilchik River. And I, as you can see, used a plunger to put the tag that sits in the Chinook salmon's stomach and then released it to migrate upstream to the spawning grounds. And since they're done feeding at this point, the idea is that this really isn't interfering with their ability to continue eating on their way to the spawning grounds. So these tags helped us locate where to go. And I used an aerial survey as well as just floating the Ninilchik River. We actually found spawning females really far up in upper reaches of the Ninilchik River. That was pretty surprising. But overall, pretty low density of carcasses further up. We found in total 23% of the female carcasses over the 2 years did have pre-spawn mortality. So they had some form of egg retention. The metric we used was at least 100 eggs were retained. And then because there were so much bear activity in these upper reaches and just in general on the river, we didn't have an ability to link heat stress levels of the spawning adult to their egg retention as carcasses on the wild. So we really couldn't make, we didn't have any evidence to detect a relationship with temperature and egg retention on wild spawning grounds. We did have enough samples though to at least look at the egg survival to the eyed stage in the hatchery. And we were able to link heat stress levels to their to the number of eggs that had survived in the hatchery program. So I used a linear model here. The response variable is egg survival. This is represented as a percentage of eggs that have survived to the eyed stage. We have HSP70 as one of the predictor variables, as well as body size as a predictor variable. Again, we used a stepwise regression approach, looking at significant p-values and removing insignificant p-values from the model. And then we did include fewer model parameters because our sample size was 39. We did a power analysis and we did need at least 43 samples to detect, to have the power to detect evidence between these relationships. But we were close enough and we decided, let's just reduce the number of parameters for this model. But what we found was that there was actually no significant covariates in the model. So looking at this plotted, we really had no evidence to detect whether or not heat stress and body size had an effect on egg survival to the eyed stage in the hatchery. So what you would expect to see potentially from the raw data, and this isn't what we saw, but just to give you an idea, we would have expected to see increasing body size to increase the egg survival to the eyed stage. So that's the left plot, x-axis being body size and the y-axis being percent of eggs that have survived to the eyed stage. On the plot on the right, you can see on the x-axis is HSP 70, and on the y-axis is percent of eggs that have survived to the eyed stage. And as HSP 70 increases, you would expect that there would be less egg survival as more heat stress would implicate their reproductive success. But this isn't what we found. So just to recap my chapter 2, looking at my my results, whether or not heat stress in spawning females affected egg retention on wild spawning grounds. We weren't able to link egg retention to heat stress, but we were able to at least get baseline numbers of what carcass surveys in Southcentral Alaska may expect in a pretty cool year. You know, a pre-spawn mortality rate of 23% is moderate. And the studies have suggested that as rates, any rate above 10% are considered elevated and could reduce abundance over 20 years. So the idea or the assumption that all fish that make it upstream past the weir and spawn fully is maybe not necessarily as we expected. Another finding from this research is that we really didn't have evidence to detect that temperature was the main driver of lowered egg survival on the Ninilchik River. So that makes us question what other drivers could be reducing egg survival there. So we also didn't see a body size influence on egg survival to the eyed stage. Just want to go through the caveats of this research now. The river temperatures were pretty average for the past 2 years we studied. And, you know, this work was really driven by 2019's high heat event, and we're heading into another El Nino year. So we might have another hot summer. So just thinking about what we saw in the past 2 years and what we can project moving forward based on these findings. This really wasn't a controlled lab experiment. We had so many factors to deal with fishing on the river, with the hatchery program, with just trying to navigate hatchery wild. And I think that we also, though, were able to actually get more of like a real life picture of what is happening on this river, on the Ninilchik and Crooked Creek. And so I'm glad that it turned out the way it did. And I think that we found some really interesting results because of it. And as you can see in this picture, this carcass was completely licked clean with the radio telemetry tag right next to it. And so we have an inkling that brown bears were interfering with our carcass survey. We even had some run-ins with brown bears that were pretty scary on the upper reaches of the Ninilchik River. And so just thinking about, where we see pre-spawn mortality rates now, could it be even higher with the presence of brown bears eating most of the carcasses that we found? Ultimately, we don't really know because we couldn't link heat stress to egg retention because so many of our carcasses had been, we'd gotten beat by the brown bears. So thinking about those future prospective grad students in the room, we have some work to carry on. We can look at the effects of moving past the effects of egg survival to the eyed stage in the hatchery. We could actually look at the fry stage as the eggs grow a little more. We can have a better understanding of how heat stress in the spawning adult is impacting offspring success. Something that I probably breezed over a little in the beginning, but I can go back to, is that there are, differences in the Ninilchik River versus Crooked Creek Raceway design. So Ninilchik River has a less space, and Crooked Creek has these big concrete raceways. And so the idea here is potentially moving brood stock in a truck from Ninilchik River to Crooked Creek, and actually setting up an experiment where we can compare egg survival to the eyed stage between the two holding areas. And that could indicate whether or not we really want to make changes to the raceway design, or if there's something else that's involved here that we don't know. And then, like I said, this summer could be a really hot summer and my research is ending. And so having some sort of heat stress protocol in place for when we have these high heat events, and we can understand whether or not heat stress is impacting those reproductive processes in the hatchery. I think this could be relatively easy, just taking some samples during egg takes and then shipping them to the East Coast and hoping for the best, hoping to see some results for the reproductive consequences piece. So some considerations for hatchery managers, we can think about changing the hatchery temperatures that the juvenile fish are reared in. So making those temperatures more variable. I know Kodiak Aquaculture Association has been having their hatchery facility mimic the river temperatures. So that would mimic what the wild juvenile fish are experiencing. And so that could be an interesting thing to explore and see how that affects the adult thermal resiliency moving forward. This is obvious. I know that it's just a challenging part of working in fisheries is we have to hold the fish as we interact with them. And so just reducing the overall holding as much as possible, thinking about when we put fish in raceways, if we absolutely need to. But this is I think for the most part, we're all trying to hold fish and make it as little stressful as possible. And then the other consideration is thinking about other stressors than temperature, since we really didn't see a temperature effect on egg survival. Potentially it's some other factor like oxygen levels in the raceway. So there's definitely more to explore there. So just to leave you today with some concluding remarks, we did detect heat stress levels above the juvenile threshold for stress, which is 2 nanograms of heat shock protein expression. We found that threshold was surpassed in 67% of the samples that we collected. So although it was a cold summer, we actually detected widespread heat stress, and we're still trying to understand what the implications of that is. We also found substantial intraspecific variation as we found differences between the populations as well as the rearing origins, and how heat stress there's quite a bit of variation in HSP 70, which we didn't really know existed. And so thinking about as variation being the raw material for selection, which can lead to adaptation and evolution. What are our fisheries going to look like as we have more heat stress and heat shock protein expression, and how is that going to define which groups do better? Again, there isn't much work that's been done in the wild. That hasn't been a controlled lab experiment where we compared hatchery versus wild rearing conditions, specifically targeting HSP 70. So our findings are novel, and it definitely opens the door to more questions as moving forward. Is HSP 70 the hero, or is it the villain? Whether wild fish have greater heat stress expression than hatchery fish, we still don't really understand whether that's bad, because at a certain level, heat stress or HSP 70 is protecting these fish during high heat events. So on the short term, it's a great solution. But on the long term, with prolonged stress, if we have weeks on end of temperatures that surpass these levels, that's when we're really going to see the differences potentially between hatchery and wild, or different rivers that are cooler versus warmer. And this is just scratching the surface of our understanding of these differences in heat stress expression. have a lot of acknowledgements to give for this work, but I'll try to go through them all because obviously the funders really made this research happen. We had so many amazing funders on this project. The Salmon Evolutionary Ecology Lab members, which is Peter's lab, they, a lot of them helped me with my defense practice talks, so many practice talks. And my committee members, Peter has been so amazing through this process. Andy and Vanessa and Steve, they, work tirelessly to get me my drafts in time. So I just really appreciate them for all that everyone has done here in this room. We have so many people that have been part of this project. I mean, KVCS from the beginning, Ninilchik Native Association gave us access to those upper reaches. We have the hatchery who picked the eggs to the eyed stage individually for this research. Cook and Lickkeeper provided the water temperature data. Mike and Holly have been integral to the work with the fish and game staff in the top right corner. They really are the dream team that started in 2019, getting me into fisheries to begin with. Kobe was my, right-hand wingman all of 2021. And yeah, it's just been such an incredible experience. And then of course, Andre, my partner, has been my rock through this whole process. My dad came and flew out from California. So thank you, Dad. We've done hunting trips with my lab, and we've gone to Seward with people from the university. And so just all in all, getting a master's has been a really fun experience. And with that, I will open up for questions, and please join us at Bishop's Beach at 6 o'clock tonight. We'll have bonfire, music, and food and drinks. So it'd be great to see you all. And thanks again for showing up. Yeah, we'll start with the online questions.
Oh.
What are they saying?
Horrible.
Oh, we have Franz, who can you?
Yeah, hi. Hey, thanks. Great talk. Can you hear me okay?
Yeah, hey,
Yeah,
Franz,
okay.
it's you.
Yeah, I was just wondering about the HSP 70 as a heat stress hormone. I guess I always just associate it with that. And you found the higher levels in fish held as broodstock. And I guess you're suggesting that it's an indicator of stress more generally. And I was wondering if there's evidence for that in the literature for fish or other species.
Yeah, a lot of the literature actually found like short-term handling stress doesn't, interact with HSP 70. And so we were thrown a little bit about that. But I do think that what we saw is just prolonged stress, as well as maturation. These fish are at the end of their lives when we sample them. So maybe that is also playing a role. I know there is unpublished work from Steve McCormick that has found greater HSP 70 in fish that are more mature than those that are not. So that could also be part of what we saw. But yeah, more of a cellular stress response rather than a thermal response there.
Yeah, thanks.
Thanks, Franz.
Maddy, maybe I follow up on Franz's question and ask what you think about the possibility that it's more of an interactive effect. Like, do you think the handling stress, could just be making fish more thermally sensitive, so you don't see the same increase because you maybe don't have temperatures so cold that you're really finding a temperature where there wouldn't be an interaction between thermal stress and the handling or something along those lines?
Yeah, I think that I think with warmer temperatures, we would see even more of that effect with potentially stress from the temperatures as well as the holding. But since it was a cooler year and we just didn't see that greater expression in other higher temperatures, we just really saw it at that 16 degrees Celsius. I think that's an indicator that there was something going on other than temperature that was influencing the the HSP 70.
Yeah, I guess what I was wondering if like, you know, if you had something like 10 or 12 degrees Celsius, like much cooler, you know, maybe you could be handled and not have an HSP 70 response at a much colder temperature than we were able to observe.
Yeah, no, I think that's a really good point. Yeah, even cooler temperatures, and so temperature isn't even a factor at all. Yeah, definitely.
Thanks.
And just so people know, we're gonna have a great period of time with the committee to be asking all kinds of questions for a bit after this. So we can follow up on some of that. That's a good question, Vanessa. Okay,
Oh, there's one here. Anna says, would it make sense to do future work that compares a stream that does and does not have raceways? And also its sport fishing pressures. So many stress variables to explore. Yeah, I think like looking at the Yukon River work, you know, they don't have the hatchery program there. They don't have as much human pressures. And so that's a good start for us to look at comparisons between heat stress levels between our systems in South Central versus that region in the Yukon. But yeah, of course, more studies are always welcome.
Gonna print that out.
that's probably the same question. Yeah. Maybe moved in on the room here.
Okay, yeah, let's open it up to the room. Bjorn.
So you were just looking at Chinook, but is it apples and oranges between metabolisms with the other salmon, or can we extrapolate anything here?
Yeah, I mean, I don't, yeah, I think that we can look at these thresholds for Pacific salmon more generally. We are seeing more declines in Chinook salmon, but sockeye salmon are doing really well right now. And so I think having more heat stress research on sockeye salmon, for example, to compare the two, I feel like so much of the research is really focused on, you know, coho or Chinook salmon, and they are generally larger and have more metabolic needs, and so probably are more likely to be heat stressed. That's my assumption. Sue.
Great talk, Maddy. So much to unpack, and I am curious about your juvenile salmon story. Correct me if I've got it right, that juveniles in the warmer stream, the Ninilchik, showed less expression than the juveniles in Crooked Creek colder system.
So we're taking heat stress samples only from adult Chinook salmon.
Oh, okay.
But we were looking at their rearing conditions. So if they're hatchery, they're reared in hatchery conditions versus wild, they're reared in wild conditions. So linking their early life history to their heat stress sample as an adult.
Oh, okay.
Yeah, sorry if that was confusing.
And I guess, yeah, I guess what I'm thinking a lot about is like this, like the terms that we use create a lot of assumptions. So if we think of heat stress, like that it's a reflection of something bad for the fish, as opposed to fish that are showing those heat shock proteins are actually showing a sign of great resilience, and that they actually are responding and protective. And that seems really like thinking about, because we're not just seeing cold, warm temperatures in the freshwater, we're seeing them in the ocean as well, right? Like the blob of 2016 is probably tied to some of our really poor returns now. And I'm wondering if there's any, I know that you did do the research in the saltwater, but I'm curious just from your understanding of the literature, do we understand if the same response happens in the ocean?
Yeah.
And would fish that are showing those heat stress responses, could we be considering them more resilient as they head out? Or, you know, like if they're in the ocean and they've got a lot of that response, isn't that maybe a positive thing?
So for people on Zoom, the question is whether or not we should be concerned about ocean conditions and whether or not, as warming ocean temperatures, is that going to be a concern for that life stage in Chinook salmon or salmon in general. And I think that because like that doc 2020 paper suggests that there's the embryo life stage and there's the spawning adult life stage that are the most vulnerable to these warming temperatures. And so I think that they are going to be those are the two life stages that are also in freshwater. And I think that that's why we're so concerned about looking at freshwater temperatures. But yeah, I mean, we can always imagine a future where the ocean is so hot that, it is causing a heat stress response. I think that's definitely possible. And what was the second part of the question again?
Well, I guess just, is the expression of heat shock protein.
Oh, yeah.
A bad thing, or is it
Yeah.
a sign for resilience?
Yeah, so is heat shock protein a sign of resilience? And I think that that's definitely something I grappled with a lot in this research and trying to understand, okay, we're seeing more heat stress in wild fish. Is that a bad thing? And I think that ultimately, at the levels that we saw them, they're sublethal levels. Like it may not be, it just may be like a subtle difference between these populations or between hatchery and wild. But I think that like I suggested in my concluding remarks, like over the long time scale. So if we're starting to see weeks on end and, adaptation hasn't occurred where they are expressing enough heat shock proteins, or they're expressing too many, like, I think we just don't understand at what level is too much. And so that's where it's going to be really interesting to see, and keeping an eye on HSP70 as a biomarker to understand this, and just having more data available. Like there's so limited comparisons across the state of Alaska. So having all this work with Vanessa Von Biela's team at USGS is just going to really propel our understanding even more. Reed?
You might have said this, but in comparing the samples you took in the two rivers, what was your sample size for each of those?
Yeah, so for Crooked Creek, it was about 40. And then for the Ninilchik River, we were looking at much more fish, like around 200. So pretty different sample size between the two. Holly?
I was thinking about your chapter 1, where you were looking at body size relation to HSP70 expression. Was that just females, or were you looking at males as well?
So the question is for body size and heat stress expression, and whether I was looking at males or just females. So for the research, I was focused on females. We did collect a few males, but we decided to just remove that from the data set because there just wasn't enough to really detect comparisons between the two. Yeah.
Yeah, one more question on that.
Oh,
From Teresa Fish.
okay. Teresa Fish, if HSP70 expression increases with progression through maturation and broodstock are selected for holding on ripeness, would you expect brood to have higher levels? Yeah, exactly. So I think that was something we really were trying to understand, is the greater heat stress levels we found in broodstock, was that due to their maturation stage, or was it due from holding stress? So we tried to account for that by looking at the day of sampling to see if, as fish move forward in their reproductive processes, are they going to be more mature later on? But, I think that, of course, fish mature at different times throughout the season. So, it's possible that we missed that as well.
Okay. It's exactly an hour. Beautifully done.
Thank you, everyone. for being
Going
here.
once, twice. Okay, well, before we give her another round of applause, just for the committee, we'll take a, say, a 15-minute break. We'll reconvene at 11:15, back on this same Zoom link. So Maddie and I are gonna go to a smaller room, but the committee will be back on the same link. And now we can clap again.
Yeah.